Good morning, class, and welcome back. Welcome to our second day of virtual learning on the plasma membrane. So the first thing you know that you need to do is you need to go to our Google Classroom and you need to do the daily attendance sign in. So make sure that you go there and sign in on the Google form under attendance for today. Once you sign in, then I want you to go to the assignment task for today and do the wellness check question. Of course, you know every day that we meet, we always do some type of activity just to see how things are going for you since we're at home learning virtually. And so make sure that you answer the wellness check question. And then at the end of the class, if you would like to meet with me or you need to talk with me about something or you want to discuss your wellness check, then just stay on the line at the end of class and we can talk about it. So please do those two things for me right now. And we'll take about 3 or 4 minutes to do that. And I will be checking the timer. So you can do that right now. And we'll wait for a few more people to come in and join our class today. All right. So I think everybody is ready now. If you haven't had an opportunity to complete it, then of course, just go ahead and do that as we're moving forward with our lesson. So of course, we always want to be reminded of the expectations for virtual learning. You know, when you come into the session that your mic should be muted, but your video should be on. I like to see your faces. And I also, I have my phone here, so I'm looking at the chat box and I'm looking to make sure that everyone is here who said that they were here. And so I am checking that. So please leave your cameras on unless, of course, you're taking a break or you need to turn it off for a short period. We want to always be respectful of the opinions and inquiries and questions of others because we're all here to learn and we just want to make sure that we interact with each other in a respectful manner. We want to engage and participate in all activities and tasks. That's part of your grade. Because we can't connect personally, we want to make sure that when we're learning virtually that you are engaged. I do my best to make the lessons engaging for you. And so my expectation is that you're going to participate. If you have questions or if I ask a question and you want to answer it, then you're going to type that into the chat box. And again, I'm monitoring that on my phone. And so I'll check it periodically throughout the lesson to see if anyone has a question. If you want to share something while we're talking, then you can type share into the chat box and I'll call on you to unmute at that time. So then when we move into our small group sessions, you're going to completely leave the whole group session, but you can always pop back in. You will immediately go to your small group session that has been pre-assigned to you in Google Classroom, and you'll join your small group. Now, if there's an issue, then you can stay back and let me know, but the expectation is that you will go immediately to your small group classroom. And as you know, I pop in and out of those classrooms to check attendance and to see if everyone is participating as they should. So I will be popping in to verify that you are present in your group. And then when it's time to return, every group has a timekeeper. So when it's time to return to the group, then I expect that your group will return within, 20, 30 seconds, no longer than a minute beyond the time. And that is the timekeeper's responsibility to make sure that you all are tracking your time while you're working. And then lastly, we want to take breaks responsibly. You may have to go to the restroom or your parent may call you. You may have to see about a sibling or whatever the situation may be. Open the door. All that I ask is that you make sure that your mic is muted and you turn off your video while you take care of that quick task or take that break. You may need to get a drink of water. But when you get ready to do that, please, I ask that you type break into the chat box so I will know that you're away. And that way, if you are away, when we move into small groups or something, then I'll know to tell you, hey, you need to go join your small group. So please just follow those expectations. They are also posted in our Google Classroom. And I think we're ready to move on. Does anyone have any questions, though? If you have any concerns about our expectations, these are the same ones that we have every day. But we just want to revisit them and make sure that you're clear on all the expectations I have for you. All right. So now it's time for your warm-up. So your warm-up should have populated in Google Classroom now. So you should be able to just toggle over to Google Classroom. And this same question is there, but I want you to answer it in the classroom under that assignment for today. So we're looking at a graph, and you know that graphing and data analysis is something that we do all the time. And so as you can see from this graph, it doesn't have a title. I purposefully clipped it out because I want to know what you would title this graph. So that's the first question. What title would you give this graph and why? So you need to look at the information that's presented. Does it have everything that you need? We've talked about that when you analyze a graph. You'll see that this graph is missing something. What is it missing? But what would you title this graph and why? And then based on the data that is presented to you in this graph, I want you to write a conclusion about the results of this experiment. So you know our process for that. The same question is in Google Classroom for you. And so I'm going to give you 5 minutes to toggle over to Google Classroom and answer the warm-up question. And then we will revisit this question at the end of this session. Okay, so your 5 minutes begins now. Go ahead and toggle over to Google Classroom. All right, guys, that's your time. If everyone can come back to the main session now. You should have answered the question in the classroom. If you were not able to complete your answer, then you can, in between time, you can work on that as long as you have it answered by the end of the session when we revisit it. All right, so our topic today, we've been talking about the plasma membrane. We've been talking about cellular transport a little bit. We got a little preview introduction yesterday, but mainly yesterday we talked about plasma membrane. Well, now we're moving into standard SB1 element D. We're talking about cellular transport and homeostasis. So of course we talked about in SB1 with that standard, you're going to have to be able to obtain, evaluate, and communicate information. So that's 3 different things that you're expected to be able to do. And you're analyzing the nature of the relationships between structures and functions in living cells. So the structure that we've been focusing on primarily for the past few days is the plasma membrane. That is the structure. And then we're looking at, well, how does it function? So that structure, what about the design and structure of the plasma membrane helps it to function in living cells? And so today, our focus is element D, which is to plan and carry out investigations to determine the role of cellular transport, examples, active, passive, and osmosis, in maintaining homeostasis. So we're going to begin looking at how cellular transport helps the cell to maintain homeostasis. And it uses the structure of the plasma membrane. So our learning target for today, we just really have one. I can design and conduct an experiment to explain how cell transport maintains a balance in cells without using energy. So that's what we're focusing on today. Now, tomorrow, our learning target will be similar, but it will be focusing on maintaining that balance while using energy. But today, we're going to simply focus on this, okay? So you see in parentheses, I have homeostasis. So that's one of your key vocabulary terms we're going to get to. But you see I have maintains a balance in cells without using energy. So that's underlined. So we know that that is a way to define homeostasis, okay? So as we progress through, then we'll highlight some of these other vocabulary words. All right, so your vocabulary terms. These are the words you're going to be looking for throughout the lesson today. And of course, as always, for this particular unit, we have an interactive word wall. And so as you encounter the terms and begin to understand the meanings of these terms, you're going to continue to update your Google slide presentation of your interactive word wall. So I've divided the words up. And so we have solution, diffusion, and equilibrium. Now we talked about the difference between tier 2 and tier 3 vocabulary words. So with these words, solution, diffusion, and equilibrium, why are those tier 2 words? And can I get someone who wants to share? If you'll type share in the chat box. All right, Brianna, if you'll unmute. Thank you, Brianna. That's absolutely correct, because those are words: solution, diffusion, and equilibrium. Those are terms that we see or hear or use in other classes besides the science class, right? But we know that with the tier 3 terms, those are words that we primarily just use in this class. So we're not really going to use those at home with our family or in someone else's classroom. Those are words specific to our class. But all of these words, you are responsible for knowing the meaning. And so as we go through and use the words and come across processes that address those, or the definitions or meanings, you will be responsible for updating your interactive word wall. And remember that interactive word wall is due at the end of the unit, and we plan to finish this unit next week. So if you're behind on that, you need to be updating your interactive word wall. All right, so before we jump into what we're going to do today, we always like to recap. So let's review what we talked about yesterday. So yesterday we talked about what structure? The plasma membrane. Right, and you'll see this diagram. We talked about components, what it's made of, and some of the features of the plasma membrane. So let's do a quick review of what we talked about yesterday. Let's see what you remember. All right. So first of all, do both prokaryotic and eukaryotic cells have a cell or plasma membrane? Type your answers in the chat box for me. All right. I think everybody's looking good. It looks like everybody has got that one for us. That is, of course, true. Yes, they do. All right. So we know that cell membrane is another name for plasma membrane, and we know that all cells have a cell membrane. Very good. All right. All of the substances listed are components of the plasma membrane except, and so look at that list. Which one is not a component of the plasma membrane? All right. I'm checking the chat box. All right. We're missing about 3 people. Okay. I think everybody's in now. All right. Let's see. Nucleic acids. So I think most of you got that one right. I saw 1 or 2 people who did not choose nucleic acids. And what are nucleic acids? We know that's DNA, RNA, right? And that's in the nucleus of the cell. But in the actual membrane, we know that when we looked at the diagram previously, that we saw the proteins, we saw the lipids, we saw cholesterol. So those are the things that comprise the membrane. All right. This one everybody should remember, because we had a big discussion about this yesterday. The feature of the cell membrane that allows it to choose what passes through it. All right. If you'll chat your answer in. All right. Everybody got this one really quickly. We spent a lot of time talking about this yesterday. Yes, selective permeability. All right. Rolling right ahead. Because of its many substances, the cell membrane is called a blank. And this is the nickname that we talked about yesterday. Go ahead and chat your answers in the chat box. Okay, I'm checking, and everybody has their answer in. It looks like maybe one person. Okay. All right. I think everybody has their answer in now. And it is fluid mosaic. We talked about fluid being moving and flowing, and then the mosaic meaning made of multiple pieces. And so we know that that is an accurate description of the cell membrane or plasma membrane. All right. So in summary, we discussed yesterday, the plasma membrane is the boundary between the cell and the outside or external environment. It helps to keep the shape of the cell, and that all cell types have a plasma membrane. So those are some of the features or characteristics of the cell membrane. But now moving into more of its function. Yes, maintaining the shape of the cell and keeping all those contents of the cell inside is a primary function, but it has additional functions as well. And so we're going to move more into that, the cellular transport piece. So how things move across the plasma membrane, because we know that our cell needs nutrients. They need certain substances in order to survive. So those things have got to be able to move into the cell and out of the cell. We talked about the characteristics of cells, prokaryotic and eukaryotic. We know they have to take in substances, and then substances also have to leave the cell. So that is cellular transport, like transportation. All right. So there's passive transport and active transport. Today, our focus is really just passive transport. We will get to active transport after day after tomorrow. Okay. So passive transport. This is when substances move across the cell membrane with the concentration gradient. Where did we see that word before? All right, chat it in. Right, it's one of our tier 3 vocabulary words. It's a very important word, and we're going to address it in just a minute. But it's important to note that with passive, when you think of somebody being passive, that means they're not aggressive, they're kind of laid back. So passive transport is transport that does not require energy. It just happens naturally. No extra effort put in for it to happen. And so in cells, there is that type of transport. And the examples are diffusion, facilitated diffusion, and osmosis. So that diffusion just means simple, plain old diffusion of substances. Facilitated diffusion, when you facilitate, that means to help. And then there's osmosis, okay? And that's diffusion of water. Now, we said the concentration gradient was one of our key terms. And so this, of course, this presentation will be available to you. This is a good diagram, I think, to use for your interactive word wall if you would like, or you can find your own. But when we talk about concentration gradient, we're really talking about when molecules are in a solution or in an area and they're not distributed or spread out evenly or equally. So if you look at this diagram, you see high concentration on the left and low concentration on the right. And then you see that wedge and the arrow. So that wedge with the arrow, that's the gradient. So what it's saying is because there's more on the left, then naturally particles are going to flow down that gradient to where there are less, to kind of spread it out. Everything in this world, all natural chemical processes typically are trying to reach some type of balance or equilibrium. And so molecules and substances tend to move to spread out where they can all have room. Okay. And so the next image you see is the arrow going up the gradient. Well, sometimes you have substances where it's fewer down here on the right, where you see there's none, because they've all moved up and they've all joined the masses. Well, that does happen as well. But which one do you think happens naturally or without effort or energy? Check that in for me. All right. Somebody said the downward. All right. Somebody says high to low. Okay. Yes. So the moving from high to low is what happens automatically. If you're standing on a hill, is it easier to stand at the top of the hill and to roll down? Or is it easier to push something up a hill? Naturally, to be at the top of the hill and roll it down. So that is without energy or much effort. So that's what we mean when we mean concentration gradient. All right, so when we have diffusion, that's the net or overall movement of particles from an area of high concentration, where it's a lot, down to an area of low concentration. So it's moving with the concentration gradient. And the result is a balance or equilibrium. And so with that, you have facilitated diffusion and osmosis. So facilitated diffusion and osmosis are examples of regular diffusion. All right, so if we look here, I know the image is not all that clear, but the link takes it to an animated image that I have uploaded in Google Classroom for you to see how the particles move. But as you can see, which side has more red dots? Well, the left side does. And the particles in the image, you see the arrow. So now they have moved across that membrane in the middle. And then on the right, you see now an image of the particles evenly spread out. So they've moved with the concentration gradient across that membrane. And that's what happens. That's what happens in diffusion. So with facilitated diffusion, to facilitate means to help. Like I'm facilitating this lesson. I'm helping you to learn. But you're in charge of your own learning. I'm just here to assist, to help, and support. So with facilitated diffusion, you have these special proteins that help or assist substances, or they create an opening or a pathway for substances to move into and out of the cell. And again, because it's diffusion, it's a form of passive transport, so energy is not required. And this is a diagram that shows this passive transport. Now, I'm checking the chat box. So if anybody has any questions, you can put those in while I go over this diagram and I'll address those. All right, so then we have the molecules that are transported, and then you see those proteins. You see a channel protein, and then you see a molecule that carries. So one creates an opening, and then one helps the substance to move. But both are forms of diffusion. One is simple diffusion, one is facilitated. Both of those are passive transport. All right, let's see if we have any questions here. All right, no questions in the chat, but remember, of course, at the end of the session, you can stay on for a review or questions if you have them. All right, so osmosis. Osmosis is the diffusion of water. So now we're not talking about just any old substances moving across the membrane. Now we're talking about specifically water moving across the membrane. And again, because it is diffusion, it's moving with the concentration gradient. So where there's least resistance, it's going with the flow. All right. And again, does not require energy. Does not require energy. Very quickly, we keep talking about this energy. Well, we don't turn on a switch for our cells to work. So just a refresher, I want to see if you remember this. What is the unit of energy that we're referring to when we talk about this energy? Chat it in the chat box. You've got 5 seconds. All right. I like it. I like what I'm saying. Everybody's chatting in. Yes, ATP, adenosine triphosphate. That is the energy that we're referring to. And in this case, it's not required for osmosis, diffusion, facilitate diffusion. All of those are forms of passive transport. All right. So with osmosis, in the previous picture with diffusion, we saw the red dots moving. The water was staying where it was. The dots were moving. Those red dots were moving. Well, here, water is represented in blue, of course, and then green represents sugar. So on this image, you see the selectively permeable membrane, though the dashed pink line, and that represents the cell membrane, of course. And so what we're saying is, instead of the green dots moving to make it balanced, the blue dots, the water is what's moving. So that's osmosis, because water is a substance that is moving to try to dilute. And you've heard of dilute before. When you make Kool-Aid and you add water, you're diluting. So the blue dots are moving. They are moving where there is less sugar on this side. And so you see the arrow moving. It's less sugar and more water on the left. And so some of that water is going to move to try to bring down that high concentration of sugar. So the sugar molecules are too big to move across the membrane, but the water molecules can move. And so that's what's happening in osmosis. It is the movement of water to try to bring an equilibrium. All right, so let's do a quick assessment. Let's see where you are. If you're getting what we're saying here. Which structures help move substances across the membrane? All right, I see you chatting in, and we're waiting on about 3 or 4 more people. All right, good job. Yes, proteins. I'm looking here. I'm seeing that I only see a couple of people that did not choose B. So we talked about those channel proteins, those transport proteins, and I showed you the image of how they facilitate in those molecules moving. And so proteins are those substances that are part of the membrane that are critical in helping substances move across the cell membrane. True or false? Osmosis does not require energy. And that is true, because we know that osmosis is what? Passive transport. All right. So energy is not required for that to happen. It occurs on its own. It's the natural flow. All right. So now we've reviewed about the plasma membrane, and we've talked about the different types of passive transport. But in addition to water, we know there are other substances in our bodies and solutions. We know there's salt, there's glucose, there's sugar, all of these substances that we've talked about in class before. We know that they are a part of what makes us who we are. They make up our body, and our cells are in these solutions constantly, right? Now, we're primarily water. 70 to 90% of our body is water. The majority of our cells is made up of water, but there are also other substances in that water. And so in our bodies, our cells are constantly inside of these various solutions made of these substances. And so there are three types of solutions. They can either be isotonic, hypotonic, or hypertonic. And so when we look at the root of isotonic, iso means same. And if you look at the definition, the first word you see is same. So the concentration of the substances in that solution are the same or equal as inside the cell. So if it's 70% inside the cell, it's 70% outside of the cell. That concentration of, say, sodium or whatever is the same. Okay. Hypo means lower. So hypotonic, a lower concentration of substances in the solution than the concentration of the substances inside the cell. So hypo, lower. All right. And then hyper, well, you've heard of hyperactive and all that kind of stuff. So hyper means, it's busy, it's going a lot of energy. So higher, faster, higher. So we see higher as the key word here. It's a higher concentration of substances in the solution than a concentration of the substances inside the cell. So when you have cells placed in these solutions, the water is what's going to move. So when we talk about tonicity or these types of tonic solutions, we know that water is what's going to do the moving to try to bring about a balance. Okay. So instead of the substances, the salt or the sugar moving, the water is going to move. But the question is, which way will the water move? Will it move in the cell? Will it move out of the cell? Or will it just move back and forth and stay at a constant rate and stay the same? And so that is the question that we have to ask ourselves. So that's what we want to know. What do you think will happen to a cell when placed in each of these types of solutions? Now, before you start typing in the chat box, let me go over this. So we've already talked about the fact that in this instance, water is going to move. When you're dealing with these solutions, water is what is moving. But what do you think will happen to the cell when it's placed in these solutions? Okay, so I'm going to do the first one for you, and then I want you to make your claim about the next two. Okay, so I'm going to look at isotonic solution. So we already established that iso means same. So that means the concentration of, I'm going to say sugar. Concentration of sugar outside of the cell is the same as the concentration of sugar inside the cell. So it's balanced. So the sugar doesn't need to be diluted or anything on either side. You don't need to lower the concentration or raise the concentration on either side. So I'm going to say that the water is just going to move back and forth at the same constant rate, but it's going to stay the same. The cell is going to stay the same size, and that's what the cell is going to work towards, staying the same size because the concentration is perfect. Iso means same, so it's perfect, it's balanced. So I think that the cell shape or size won't change and that the water will just move back and forth at the same rate. That's my claim. Now, of course, my claim doesn't have to be your claim. Now, most of you are probably going to pick the same claim because you think that I'm always right, but I could possibly be wrong. But that's what I think. So now I want you to focus on number 2 and number 3. Now, if you need to look back at the definition of hypotonic and hypertonic solution, I'll go back to that for just a second. But I want you to chat in the chat box what you think will happen to a cell when it's placed in a hypotonic solution and a hypertonic solution. Now, when you chat in, instead of having to type all of that out, you can put 2 and then write your claim, and 3 and then write your claim. And if you would prefer to type out the words, then you can, just for the chat box. Now, when we get to our next assignment, you'll have to type everything. But just for the chat box, put a claim for 2 and a claim for 3. Now, that's going to take a little bit of critical thinking. So I want you to take your time. I want you to think about what the question is asking, How is the cell going to change? What is going to happen to it in a hypotonic solution and in a hypertonic solution? So think back to how I thought through it and how I explained it in my example with isotonic. And then I'm going to take you back to the definition. So hypotonic means concentration of substances in the solution outside of the cell is lower than it is inside the cell. So you got less sugar on the outside than you do on the inside of the cell. And then hypertonic, a higher concentration, meaning more sugar outside of the cell than the amount of sugar inside the cell. All right. So go ahead and start chatting your claim in. What is going to happen to the cell when placed in a hypotonic solution versus when it's placed in a hypertonic solution? All right, I'm going to give you about 3 minutes to come up with your claim. And I'll toggle back and forth. What do you think will happen to the cell? And remember, you're focusing on 2 and 3 for your claim. I gave you 1 as my example. All right, some of you are starting to put your chat your claims in. All right, you got about another 30 seconds. All right, so you know the session is always recorded, and so I have your claims here. Now we're going to look back at your claims once we've had a chance to conduct our experiments, okay? So now you have your claims, and so now we'll move to the next phase. So now you're getting ready to move into your groups. So your groups are already pre-assigned in Google Classroom for you, as always, when you get ready to do group work. And so when you leave, remember, once you leave this session, you need to leave the session entirely, and then go into your group session in Google Classroom. Click on the Google Meet link for your group, and you should only be able to see your assigned group, no one else's. Go into your group, and then when you get into your group, remember to go ahead and you let everybody know what your role is, because I've already assigned you your role in your group. And as a group, this is what you're going to do. You're going to share your claims with each other. So that means you're going to discuss with each other what you came up with for your claim. So make sure you remember what you said. And if you need to, jot it down. And then you're going to, as a group, work through your different claims and come up with one. Decide who has the best one or combination of yours, and come up with one claim for each type of solution. So you gave a claim for hypotonic and hypertonic, but you also have to do a claim for isotonic. Now, it can be the same as the claim that I gave, or you may have a different one, but you have to have a claim, one claim for your group for each of the 3 types of solutions. What will happen to the cell, and explaining which way the water will move. All right, so you have 10 minutes to come up with your claim. So timekeepers, 10 minutes for that. Then you have to set about the task of designing an experiment to test your claim. How could you test your claim? So I've already uploaded your Google Slides template that has all of the areas for your experimental design. And I've given you a list of materials that you can use to design your experiment. So you may or may not use all the materials. That's up to you and your team. But there's an entire list of possible materials that you can use. And there's a design template for you, which is the Google Slides presentation. All of that is under your assignment for your group, okay? And then I want you to make sure that as you go through it, that you're working as a team, and it's not just one person's idea for the experimental design. This is everybody coming together. And this is not the first time we've done an experimental design. We've gone over this in depth before, and this is your 4th time designing an experiment. So you should be getting pretty good at this now. All right, so you have 30 minutes to work on your experimental design. Now, I will be checking in on each group randomly. You need to make sure that you list the role of each person so that we make sure that everybody's doing their job. I have a list, but you need to make sure that you're clear with each other about what your role is in the group. And I'll be popping in and out of the groups to check on your progress and your work. And then we'll come back together for our closing session. All right. So if you have questions while you're working in your group, then of course hold the questions until I come into your group. If you have a question after I've already been in your group, then hold it until we come back. And remember, your group leader is the one who will ask the question for the group. All right, a reminder of group roles. They've already been assigned. When you go into the into the classroom and you click on your group, you will see what your role is. You have a group leader, and you know your job. You are the one that communicates with me. You ask questions for representative of the group. You review the instructions or directions for the group once you get into the lab, or you review the instructions that I've given you. You make sure that the group stays on task. If y'all start to, playing, or somebody starts talking about something they shouldn't, you bring the group back in and keep them on task. And of course, everyone in the group does research and experiments. Excuse me. Now, the researcher. You break down the research task and you assign the group members the research task, but you are the primary researcher. So you have the primary job of research and making sure that all the research is done and that you cover all the areas. And of course, you also conduct the experiment. The recorder. You'll type, record information and data, and create slides presentation. Excuse me. And you also, of course, as a recorder, you still do some of the research and you experiment. And then the resources manager and timekeeper. You track the use of the resources, whatever resources you're provided, if it's websites, links, in this case, if we were face-to-face and you had actual real-time materials, you would keep track of that. You keep track of time. Now, I told you you had 10 minutes to work on the claim, 30 minutes to actually design the experiment. So you have to make sure the group knows what time it is and what time to come back and join the whole group. And then again, you are a research and experimenter as well. So those are your roles. You already are familiar with those. And you know that every time we meet, so whatever your role was last time is very likely a different role this time. And this is our 4th time doing one of these labs. So that means that you are probably doing something, you've done everything else, and then this is the last role, and then it'll start to cycle back again. So just make sure you understand your role. If you have questions, of course, by all means, stay back and ask those questions before you move into your group, or you can pop back over before I start going around to check in each group. All right, so before we move into our groups, does anyone have any questions about the task or the assignment? And of course, everything about the assignment is in your Google Classroom. So when you go in, you'll be able to see all the details of the assignment and your expectations for the design. Your rubric is also in the classroom along with the template for your experimental design. And then of course you have 30 minutes, but we know that if you don't finish it, we can pick it back up tomorrow. But today you are only allotted those 30 minutes. All right. All right. Let me see if we have any questions. All right, Alex. So Alex wanted to know, I think I just mentioned this, but he said he doesn't know if 30 minutes is going to be enough time. And again, you'll use the 30 minutes and you'll get as much done as possible. You'll submit what you have so that I'll be able to review it. But then if you're not finished, that's fine, because when we come in tomorrow, you will be allotted some time to finish your design. All right, before you have to present your experimental design. But try to get as much as you can done and working as efficiently as possible, and that comes from working as a team. All right. I don't see any other questions. So now we're ready to move into our groups. So remember, you're going to log completely out and your timekeeper is going to keep the time, and then you'll come back in. All right, let's go ahead and go into our groups in Google Classroom. And group 4, I see Donovan is on a break. He had to go to the restroom. So when he comes back in, then I'll send him over to your group. Okay, Donovan, I see you're back. Yes, you are in group, so you can go ahead and go into Google Classroom, and your team leader or group leader will refresh you on what the expectations are for this task for your group assignment. This is what we went over. You're going to come up with your claims based on as a whole group, and then you're going to begin designing your experiment to test your claim. So you have a total of 40 minutes, 10 minutes to work on the claim, and then 30 minutes on your experimental design. All right, so go on in your group and make sure you see your group leader, and they'll refresh you and go over everything with you if you've missed anything, okay? Thank you, Donovan. All right, everybody's back. That 40 minutes, I know it went by quickly. I'm glad to see that everybody made it back to their group one time. And so now that you've had a chance to design your experiment, then let's preview what we're going to be doing tomorrow when we come to class, because we have about 6 or 7 minutes before class, before this session ends. And I want to keep in with the time that we've allotted for the online session, okay? So tomorrow when you come in, we're going to review just a little bit. We're going to share your experimental design. So we'll review, and then I give you a little bit of time to finish up your experimental design. But of course, you know that you can work on this outside of this session. That's up to you and your group when you work on it. But you are going to share your experimental designs tomorrow. And then I will give you about 10 minutes at the start of the class to finish them up and come up with your final presentation and how you want it to look. And then we're going to go ahead and start our virtual lab to test our claims. So the virtual lab will be over osmosis and diffusion. I'm not going to tell you what the source is. You'll see tomorrow, but we are conducting a virtual lab. And that virtual lab is going to put your claim and your experimental design to the test, because you're going to compare the design that you came up with what you actually do in the lab. And then you're going to record those results and see if the results support your conclusion, okay, or support, your claim. So we'll start our virtual lab. I'm not sure if we'll have time to finish it, but the way I have it planned out tomorrow is you should be able to get it started during the virtual session, and it may run over, but then you would just complete that for homework if we don't have time to finish it in our virtual session, okay? So that's what we'll be doing tomorrow. All right, so now as we get ready to close, your ticket out of the door is in the Google Classroom. And what I want you to do, I told you at the beginning that for the warm-up, we will revisit that question. So I want you to revisit the question. And I want to know, is based on what we've done today. We've looked at passive transport. And if you'll notice, our warm-up covered facilitated diffusion and simple diffusion, which is part of what we talked about today. So now that we've had a chance to learn a little bit more about this passive transport and diffusion, I want to know, in your ticket out of the door, would you change your analysis of this graph? Remember, you had to come up with a conclusion based on what you saw. So on your warm-up, you came up with the conclusion and you came up with a title. So now that you've learned a little more about passive transport and you've learned about diffusion and osmosis, would you change your analysis of the graph? And you have to explain why or why not. So if you would not change it, you would leave it the same, then why would you leave it the same? What do you feel was right about it? And then if you wouldn't change it, if you would change it, then why would you change it? What changed your mind? What is different? Or what insight did you gain from what we did today that will make you change your response? Okay? So I'm going to close out the session, but of course, I'm going to stay back. And if anyone, we have about, I'm going to stay back for about 30 minutes. And I'll give you about 5 minutes to work on your ticket out of the door. You can answer that in Google Classroom. But if you plan to stay back, please in the chat box, put stay. Just type stay. And I'll know how many people are going to stay back with me. If you have questions, something I need to review with you. It could be something from today or the other day. But if you need me to stay and speak with you, then just type stay in before you go in to do your ticket out of the door. And then you have about 5 minutes to come back, and I will remain in the classroom so that we can review. If you need to go back over something we did today or need some support or help with something, just let me know. Okay. So now I'm going to ask everyone to go ahead and toggle over to the Google Classroom and complete your ticket out of the door. And of course, that has to be completed because I will be checking that once my after session remediation is over. So make sure that you get that done. And if there are no other questions or anything else, I'll hang back, you do that, and then you can log off if you are done, and I will see you tomorrow. So just make sure that you're ready to go tomorrow. And of course, I will upload recording of this session to our Google Classroom as well. All right, I've enjoyed you guys. I think we've had a great day and a great session. Thank you for your participation, and I will see everyone tomorrow.